Today we are going to explain the mechanism of cytotoxicity of the cytotoxic T lymphocyte. It's document 7, specific cell-mediated immune response in chapter 7 in the Lebanese program for LS students. Let's start with a quick recap about the activation of cytotoxic T lymphocytes after the antigen recognition and the help of helper T lymphocytes that secrete interleukin 2. The cytotoxic T lymphocyte is activated. It multiplies to give a clone of identical cytotoxic T lymphocytes that differentiate into effector cells and memory cells. Memory cells preserved for many years, and effector cells that kill the infected cells or the cancer cells or the graft during the effector phase. The cell-mediated immunity is effective, is very efficient against intracellular bacteria and viruses that are inside the host cells and are inaccessible to antibodies. Also, the cell-mediated immunity is triggered against cancer cells or transplanted tissue. Now, how do T-C cells attack their target? We have a double recognition. As you can see here, we have the target cell that can be either an infected cell with a virus or a cancer cell expressing MHC class I molecule on its surface. This molecule is associated with a peptide, which is a non-self peptide. So the TCR of the cytotoxic T lymphocyte is specific to the epitope presented in the MHC class I molecule. It's the double recognition. The cytotoxic T lymphocyte recognizes the non-self peptide plus the MHC class I molecule expressed on the surface of the modified self. As you can see in this picture, the cytotoxic T cell has TCR receptor on its surface, and inside the cytoplasm, the TC cell has granules, vesicles that contain toxic substances called cytotoxins. Cytotoxins means substances toxic to the cell, substances that kill the infected cells, that destroy the infected cells by the degradation of its DNA. The two types of cytotoxins present in the cytoplasm of T lymphocytes are granzymes and perforin. The cascade of cell lysis by cytotoxic T lymphocytes begins when a cytotoxic T cell recognizes an infected body cell or a modified body cell, and when it binds to the HLA class I peptide complex on the target cell membrane through its TCR. It's the double recognition. Following the double recognition, the TC releases first perforin. So the first cytotoxin to be released is perforin and not granzyme. Perforin assembles into polymers that form a hollow channel through the target cell membrane. It's the polyperforin channel. This channel will help the granzyme to migrate towards the cytoplasm of the infected cell. So the first step is the release of perforin to create a polyperforin channel through the target cell membrane. Then the TC cell releases granzymes that penetrate into the target cell through the polyperforin channels. In green, these are the granzymes. They are now in the cytoplasm of the target cell. Now the granzymes will activate or trigger an enzymatic chain reaction. The granzymes activate many enzymes in the cytoplasm, and these enzymes, activated by the granzymes, will degrade the DNA of the target cell. Thus, the target cell dies by apoptosis. Apoptosis, it's the death of a cell by the degradation of its DNA. And finally, when the target cell dies, the TC cell detaches from it and recirculates to kill other target cells carrying the same HLA class I peptide complex. And the dead target cell is eliminated by phagocytosis by the phagocytes, which are macrophages or neutrophils. So it's the cascade of elimination of infected host cells by the cytotoxic T lymphocytes. First, we have perforin release to create a polyperforin channel through the membrane of the target host cell. Then granzymes are released by the TC. They pass through the polyperforin channel. They activate several enzymes in the cytoplasm of the target cell in order to degrade the DNA of this latter. Now the target cell is dead, and the TC cells are free, are ready now to kill other target cells having the same HLA class I peptide complex. As you can see here, this is the TC, and this is the host cell, the target cell. The TC approaches the target cell, binds to the HLA class I peptide complex, and releases its content, the granules, the granzymes, the perforin, in order to lyse or to kill the infected cell. In the next video, we will talk about the relation between the cell-mediated immunity and the cancer. Thanks for listening.